All right, I'm going to get your notes passed out to you. So this is our photosynthesis overview. We're basically learning about all the major players. Tomorrow we should be able to do a plant pigment chromatography lab. I'll have my bone marking test set up in the lab, so we'll probably have to do it in here, but I think we can make that work. All right, let's try it. All right, so you don't have this slide. This is just your photosynthesis overview, but you do have this slide. So first we need to know what kinds of organisms can do this, because it is not just plants. All right, so let's take a look at the kinds of organisms that can do photosynthesis. Of course, we've got plants. We've also got multicellular algae, such as kelp. Whenever you hear the term algae, algae are the ancestors of plants, and they are protists, okay? But they can be unicellular or multicellular. We have some unicellular protists that are photosynthetic, such as euglena, and we'll see those living specimens of those under the microscope in the spring. When we get to protists, I try and order live amoebas, euglenas, stentors, paramecia, and volvox, so that it's not just looking at a stained slide, you actually get to see those moving. And then we have blue-green algae, okay? So blue-green algae is sometimes called cyanobacteria, but they are also photosynthetic. Next, I know I need to write this one word bigger. This word that's really teeny tiny is mesophyll. So you need to understand the hierarchy of what is part of what, or what is inside another structure. Get this done. Then I'll explain this slide. And so we'll watch a little animation in just a couple minutes that will kind of take you kind of through this, these levels. So you've got the leaves, and then this is a cross section of a leaf. So this is like you're looking at the leaf sideways. And all of these cells are called mesophyll cells. They are going to be the cells that carry out photosynthesis in a plant stem or a leaf. So those are not chloroplasts. Those are mesophyll cells. But the green dots that you see inside those cells, those are chloroplasts, okay? So these little itty bitty dots that you see inside them, those are chloroplasts. And so today we're also going to look at chloroplast structure. We're going to label the different parts of a typical chloroplast, but understand you have the leaf, and then you got to think, okay, in what part of the leaf are my photosynthetic cells? Well, we're in the mesophyll layer. You've got the top layer of mesophyll, which is called palisade, because they kind of look like books lined up on a bookshelf. And then you've got the ones that are more spaced out because they're closer to where the gas exchange occurs. So that's your spongy mesophyll, because they have more space in between them. But the mesophyll is the section of the leaf where the photosynthetic cells are. Within those photosynthetic cells, you will have organelles called chloroplasts, which contain pigments that carry out photosynthesis. All right, so let's look at the structure of a chloroplast. So that's what this entire thing is. Notice that it does have a double membrane. It's got an outer membrane and an inner membrane, in addition to the thylakoid membranes that surround each of those little green discs there. Remember that chloroplasts have their own DNA and their own ribosomes because they used to be their own little self-sufficient prokaryotes. Each of these discs, and I can write that bigger in case you can't see that, but each of those discs is called a thylakoid. And so those thylakoids are surrounded by a thylakoid membrane. And then there's a space inside each thylakoid, and ironically, that is called the thylakoid space. Now, why do we care? Well, same reason you have to care about the parts of a mitochondria, because we're going to talk about different things that happen there. In other words, the light reactions that we're going to talk about over the next 2 days, they take place on the thylakoid membrane because there's actually proteins and pigments that are embedded in that thylakoid membrane. The thylakoid space is where we're going to build up an H+ concentration, okay? And then this area outside the thylakoids, which is called the stroma, that is going to be where your Calvin cycle takes place, or your light-independent reactions. And so you've got to be familiar with the anatomy of a chloroplast to understand where these reactions happen. Notice that these thylakoids are all stacked up, and so a single stack is called a granum. That's if I'm just talking about one stack of thylakoids. If I'm talking about multiple stacks of thylakoids, we would call them grana. So that is the plural form of granum. And then you got to make sure you don't get this next term confused with cytoplasm. If we were showing this chloroplast in a cell, cytoplasm would be out here. Okay, this next blank, we're looking at the fluid that kind of fills up the inside of the chloroplast. It's the fluid that surrounds the thylakoids, but it's in the chloroplast. So that is called the stroma. And again, that's going to be where the Calvin cycle or the light-independent reactions take place. And then I did find a cool animation, kind of like that when we talked about microtubules. and it was called The Inner Life of the Cell. I found one that's kind of like that, but with chloroplasts. We're going to do one more slide, and then I'm going to show you that animation, or at least part of it. It's only like 2 minutes long. Because like I said, I need you to understand the hierarchy of a leaf, mesophyll cells, chloroplasts, thylakoid, like what's inside what. So what you'll see when this video shows you entering the leaf, gases enter the leaf through pores called stomata. And so stoma is singular. You hardly ever hear it called that. Stomata is plural. These are usually found on the underside of a leaf, because if they were found on the top surface of the leaf, you'd have too much water vapor evaporating through them, because that's what the sun shines on. So we usually find these on the underside of a leaf. And so the stomata, or the stoma, is actually the hole. And then on either side, you notice in this diagram, you have guard cells. So they guard that opening. And so to me, this is an electron micrograph. It kind of looks like a little mouth. And so if this was the opening to your mouth, this would be your top lip and your bottom lip. The lips would be like the guard cells, and the opening is the stoma or the stomata. We do a lab in the spring where it's with our transpiration lab, where I have you take an impression of the underside of a leaf just using really good, like shiny Scotch tape. And we use clear nail polish to kind of get the impression from the underside of the leaf. We put clear tape over it. We peel the tape off, and we put that tape directly on a microscope slide. And you're able to count and figure out the density of the stomata in that particular kind of plant. So let me go back to this video. Like I said, it won't take very long, but I need you to kind of have a visual with this chloroplast. Let me reset this. So read the captions. They're approaching a redwood tree, and they tried to make this as scientifically accurate as they could. Notice they are going from the underside of the leaf, because that's where your stomata are. So there's all the little stomata. You can see them. There's an open one with its guard cells. And now these are cells. Those aren't chloroplasts. Those are cells. And they're calling them palisade cells. That's just another name for mesophyll cells. So that is one mesophyll cell right there. And it tells you the purple blobs are the mitochondria, because remember, it's not that plant cells have chloroplasts and human cells have mitochondria. Plant cells have both. And now we're about to go inside the chloroplast. So there's all your thylakoids. And notice all the stuff that's stuck on the surface of them. Those are all different proteins and pigments that are embedded in those membranes. So now we're in the nanometer scale. We'll see ATP synthase more either tomorrow or Friday. I'll stop it there because we're not getting into the light reactions, but I at least wanted you to see that chloroplast structure. I think that's kind of cool. All right, next we are getting into light and wavelengths of light. Okay, so electromagnetic energy, light is a form of it, the visible light spectrum. This type of energy travels in waves. Okay, so wavelength is the distance between the crests of those waves. So this may not be something you've done in a while or maybe ever, but we say that that light travels in waves. When we're talking about the wavelength, we're talking about the distance between the peaks. Okay, so that little double-ended red arrow that I just drew in, that would be the wavelength. And so why do we care? Well, because we'll talk about things that have a longer wavelength or a shorter wavelength. So we'll contrast something like this with, maybe something like this. Okay, so the wavelength on this one on the upper right is going to be much shorter. Okay, it's only that long. And you can kind of tell by looking at it. If I asked you which one had more energy or had higher energy waves, which one would you tell me, the right or the left? It's the right. Okay, it's going up and down, up and down, up and down, up and down. Very short wavelengths, higher energy. Okay, so that's why we care about wavelength. And so don't flip your page yet until we look at the picture on the bottom of this slide together. Okay, so the electromagnetic spectrum is the entire range of radiation. The part we care about is the visible light spectrum, and that's from about 380 nanometers to 750 nanometers. And so all this is showing you is that the violet end of the spectrum is a lower number, so that's a shorter wavelength, which means that individual waves, they are in higher energy because the waves are come so close together. Towards the red end of the spectrum, that's a higher number, so that's more like this picture. We have longer wavelengths, okay, but that's lower energy because the waves don't come as close together. Now, really, we don't really care as much about the red versus the purple light. What we care about is which wavelengths of light a given pigment can absorb the best, because that's what's going to determine its rate of photosynthesis. People always want to look at this and be like, oh, well, if it's absorbing violet light, that's more energy. That's just the wavelength of light it absorbs. How much of that wavelength, like what color light you're shining on that plant and what color light that pigment can absorb, if it's absorbing a lot of light, yeah, it's going to be able to do a lot of photosynthesis, but it doesn't really depend on this end versus that end. I hope that makes sense. That'll become a little clearer when we look at absorption spectrum. All right. So pigments. So pigments are going to be substances that absorb visible light, and those pigments will be found on these thylakoid membranes. And that second bullet there is more important than you probably realize. Okay, it's a little bit about absorption and reflection or refraction. You don't see the color that the pigments absorb. Okay, so what we're going to look at over the next few slides is we're going to look at the different wavelengths of light that different pigments can absorb the best. And if they absorb a lot of that wavelength of light, you're going to see a pretty big peak there. Okay, but understand, if they absorb that wavelength of light, it's like a paper towel absorbing your Gatorade that's spilled. Okay, if it absorbs it, you're not seeing it because the paper towel gets rid of it. Okay, what you're seeing color-wise is the color that gets reflected or transmitted. So the plant is going to show the colors or wavelengths of light that do not get absorbed. Okay, and so what you're going to see is that green is a wavelength of light that a lot of our pigments don't absorb very well. And so because they don't absorb the green light very well, that green light gets reflected, and that's why most plants appear green. Okay, and you'll hopefully just keep that in the back of your mind as we look at specific pigments and their absorption spectra on the next few slides. So there are 3 pigments that we're going to talk about in notes today. There are other pigments that we'll get to as we start to talk about fall leaves and why they turn the colors that they do, and how weather impacts the changing colors of the leaves. So the 3 pigments that we'll be talking about are chlorophyll A, chlorophyll B, and carotenoids. So hopefully we'll get to do this plant pigment chromatography lab. I don't think I got to do that last year because it hadn't come in from the science company that we order our stuff in. So I don't even think I got to do this lab last fall. But basically what we do is they send us 2 different kinds of extracts. They send us spinach extract, and then they send us blue-green algae extract. I'm not even sure that I'm going to use the blue-green algae because the spinach works so much better. And basically what you're going to do is if you've ever done chromatography, like maybe the chromatography in the colors found in black ink, like sometimes they do that in a chemistry class, so you get used to what chromatography is. Chromatography uses some kind of solvent that helps separate out the pigments in something. So if you've ever put like a black dot on a coffee filter and put it in some solvent, I don't know what kind of solvent they use, as the solvent gets pulled up the chromatography paper, it will separate that black ink dot out into the colors that make it up. And so that's what we're going to do with pigments tomorrow. We're going to place some spinach extract on this special kind of paper and then put it in a solvent. And as that solvent works its way up, that special piece of paper, it's going to separate out into the pigments found in that spinach extract. So these will be some of them. Okay, so we're going to talk about each one. I'm going to show you what chlorophyll A and chlorophyll B look like. So for each one of these, I'm going to tell you about what color they absorb, what colors they appear, and then what their function is in a chloroplast. So this is chlorophyll A. This is what a molecule of chlorophyll A looks like. Notice the only difference structurally between chlorophyll A and chlorophyll B is that chlorophyll A has a CH3 group or a methyl group there. Chlorophyll B has a CHO, which is probably a carbonyl group in that same location. But that's really the only structural difference between the two. So chlorophyll A absorbs mainly blue, violet, and red light the best. It is the main pigment involved in the light reactions. And so if you've watched any of these, they had a college like trivia tournament where they had like 3 people from a given university. So it might be like Duke versus Chapel Hill. And these were, it's basically like an academic team kind of thing. And, for those kinds of team, quiz bowls or trivia tournaments, a lot of times you can buzz in and answer by yourself. But then sometimes there's a bonus round where you get to confer with your teammates and decide on an answer, and the captain speaks for the team. So that's the way I kind of think about these pigments, is kind of like if you're in a trivia tournament and you have a captain and then you have people who are knowledgeable in different areas. Did anyone do academic team here ever, or maybe in middle school? When I started high school, I had just moved to South Carolina from New Jersey, and I didn't know a soul. I moved down here 4 days before I started my freshman year in high school at my first public school ever, because I had gone to a really small Catholic school where there were less than 20 people in my class. And I'd been with most of those people since kindergarten. So it was like culture shock. So I'm like, I like Jeopardy, I like trivia, so let me do this. And I was pretty good on academic team until they started really strategizing for it like they do now, where they'll say, okay, Grayson, you're going to be our science person. And Alexis, you're going to be our lit person. And John, you're going to be our art person. And Noah, you're going to be our history person. And then they give them all these lists and stuff to memorize in their subject area. That seemed like extra homework to me, and I really wasn't going to do that. So that's kind of when I stopped being on the traveling team and was just a member. But I want you to think about that strategy. So if we have a literature person, and we have a science and math person, and we have an art history person and a regular history person, we have people in all these different areas. Why do you think they do that?
Everybody's a master of their area.
Right. And so especially there should be somebody on your team that can answer that question, right? You're trying not to let any questions go unanswered is kind of how you want to think about it. That's the way I want you to think about these pigments. Each of these pigments, and there's more than just the 3 we're going to talk about here, each of these 3 pigments is able to absorb different wavelengths of light. Because imagine if chlorophyll A were our only pigment. We'd only be absorbing blue, violet, and red light, and we'd be letting all those other wavelengths go to waste. We don't want to do that. That's wasteful. We want to have different pigments that can absorb different wavelengths so that collectively we're not really letting much in the way of light go to waste. Okay? So that's what we mean. Oh, sorry, I got to explain the main pigment. The main pigment would be like our team captain. So what you're going to see tomorrow and Friday is that it's kind of like when your trivia team gets to confer or talk about an answer, and then the team captain reports it. These other pigments will absorb whatever wavelengths of light that they absorb best, but then they will pass that energy onto a molecule of chlorophyll A that is centrally located in a place called the reaction center. So chlorophyll A is kind of like the team captain. Other pigments can absorb whatever wavelengths of light that they absorb best, but they will ultimately pass that energy back to a main molecule of chlorophyll A in some place called the reaction center. And so it's going to appear kind of a blue-green color. Now this stuff down the bottom, the picture on the right, I just drew in. It's not bad. It's pretty close. That's called an action spectra. Whereas this is an absorption spectra. This is what I was telling you is a lot of what your homework's on. So pay close attention here, especially if you've never ever seen anything like this, which I imagine you probably haven't. Okay, so notice what's on the y-axis, absorption of light by the different pigments. And then you just have your electromagnetic spectrum down the bottom, your visible light spectrum. So where the peaks are, that is how much light that pigment can absorb of that wavelength. Okay, so if you look at chlorophyll A, it has a peak somewhere around 430, 440. It can absorb a lot of light there. And so the thing I want you to think about is, well, what if I had that pigment? I had chlorophyll A, and I shined light on it that was 430 or 440 nanometers. Would I be able to do a lot of photosynthesis or not much? Should be able to do a lot, right? Because I'm shining that wavelength of light on it that I know it absorbs a lot. What if I shine a light on it that was 550 nanometers? Am I doing much photosynthesis? No, because that pigment does not absorb wavelengths of light at 550 very well. And so if you're not absorbing the wavelength of light that is being shown on you, you're not doing much photosynthesis. So it doesn't really have to do with whether it's purple, whether it's red. It has to do with this and how much light of that given wavelength that pigment can absorb, and then also what wavelengths of light are you shining on it. Chlorophyll B that we'll do next shifts a little bit towards blue, so it absorbs blue light pretty well, and it absorbs orange light pretty well. Carotenoids are going to absorb kind of your violet, blue, purple, and then they're going to reflect your yellows and oranges. And so carotenoids, like the word carrot or beta carotene, you think yellow and orange, they don't have any sort of peaks on this end of your spectrum, so they're going to reflect those colors. They're going to appear your yellow-orange colors. Okay, so that's the absorption spectra of those three pigments. Again, don't rely. The color of the lines, yes, it is showing you what color they appear. We set up here that chlorophyll A was going to be blue-green, and indeed it is. I know it can't be. I know it can't be purple or blue because it's absorbing that wavelength. I know it can't be red because it's absorbing that wavelength. So think about a paper towel that absorbs like Kool-Aid. If it absorbs that Kool-Aid, you don't see the Kool-Aid. If that wavelength of light gets absorbed, you're not seeing it. You are seeing the colors it doesn't absorb because that's what gets reflected. Okay, when we look at this action spectra on the right, all it is, and that's why I just freehanded it, is if I took the absorption spectra and just drew lines around all the peaks collectively. This is individual pigments. This is, okay, what do all my pigments absorb together? That's what an action spectra is. It's just as if I kind of outlined where all those peaks are. There's that. All right, chlorophyll B. Chlorophyll B absorbs mainly blue and orange light, and you should be able to see that on the absorption spectrum that's on the bottom of this slide. It is an accessory pigment. So it's not the team captain, but it is on the team and it has a special or a couple specialized wavelengths of light that it likes to absorb. Okay, so it passes energy on to chlorophyll A and broadens the spectrum of light that a plant can use. Because remember what we said chlorophyll A absorbed? We said it absorbs blue, violet, and red light the best. Well, this is different. This is blue and orange light. So you have different specialties, right? Different areas of concentration, or in this case, different wavelengths of light that it absorbs, because you are trying to maximize the amount of light that the plant can take in. Like we said, chlorophyll A is going to be the main pigment, so chlorophyll B will absorb whatever it's going to absorb, and then it's going to pass that light energy to that main molecule of chlorophyll A that's going to be located in our reaction center. And then finally, it is going to appear more of an olive green, which you can kind of see in that absorption spectrum picture. Make sure you can see how this first bullet is shown on this absorption spectrum graph. We've got a peak over blue, we've got a peak over orange. You don't have to memorize. I mean, I kind of know that the low numbers are like violet and the high numbers are red, but the question like this that they've given on AP exams, they've given you a chart where it lists like green is found between this wavelength and this wavelength. So you don't have to memorize stuff like that. All right, then we have carotenoids. So carotenoids, think carrot. They are going to appear yellow and orange. And I know, remember, if something appears that color, it doesn't absorb those colors. It reflects those colors. So we know that this pigment is not going to absorb yellow or orange wavelengths of light very well at all. Instead, it's going to absorb violet and kind of like a blue-green shade of light. And these serve a protective function. Because they tend to absorb violet light, remember we said that the violet end of the spectrum, it has shorter wavelengths, so they're higher energy. And those are wavelengths that could be potentially damaging to the plant. And so the carotenoids are able to take in those violet wavelengths and sort of break them up so that they don't harm the chlorophyll. Okay, so you see that it kind of has this peak down where you have shorter wavelengths but higher energy. And so basically carotenoids aren't harmed by those waves. And so they absorb them and they break them up so that they don't do any harm to the chlorophyll A and chlorophyll B. We have no absorption peaks over here for carotenoids. So that's why they are going to reflect and appear those yellow, orange kind of colors like we see in a lot of our fruit. So three main pigments are chlorophyll A, chlorophyll B, and carotenoids. In your lab tomorrow, if we do that plant chromatography lab, we'll have xanthophylls, anthocyanins, stuff like that. We'll talk about additional pigments. All right, so where would these pigments be found? Well, a collection of pigments is called a photosystem. And these photosystems are going to be embedded or stuck in the thylakoid membrane of the chloroplast. So a photosystem is a cluster of pigments embedded in the thylakoid membrane. Now, just like any other membrane, this is going to be a lipid bilayer. And so this will be a cluster of pigments in the lipid bilayer, specifically the thylakoid membrane. We've got two photosystems. You don't really have to memorize the wavelengths that they absorb best. I can't always keep them straight. Basically, it just has to do with like averaging which pigments they have, and that's kind of the average wavelength that they tend to absorb. But notice you have, even though we tend to draw all these pigments or color them as being green, it's not all chlorophyll A and B. That's why I put this picture in there. You've got one called phycoerythrin. Okay, erythro means red, like an erythrocyte is a red blood cell. So that is a red pigment. You know our carotenoids are going to be yellow-orange. You've got xanthophylls, which I believe are yellow. So I don't want you to think that pigments are just green like you see here. But the reason I put this picture in there is because it's showing you, okay, we're looking at that thylakoid membrane here. We can look at that picture and say, oh, I know that's a membrane because it's a lipid bilayer. This thing in the middle is the photosystem. And then each of those little ovals that you see is a pigment. And even though they're all shown as green, they are not all chlorophyll A. They can be chlorophyll A, chlorophyll B, carotenoids, phycoerythrin, xanthophylls, anthocyanins, all these different pigments. Okay, this might be a little over our heads today, but I at least want to expose you to it. This will help you out with what we're going to do tomorrow. Okay, so a photosystem, we know a photosystem is a collection of pigments, but there's also some other components to it as well. There's a part in the center called the reaction center, and it has chlorophyll A and then something called the primary electron acceptor. And then the part where the pigments are, those are on the sides. And so each of those little green dots there, that's a pigment with a main molecule of chlorophyll A in the middle. And so these are the light harvesting complexes that are shown in that darker purple. The lighter lavender color is the reaction center. So you don't have to understand everything about that today, but that will play a role in what we do tomorrow. So it would be helpful to understand that a photosystem is a collection of pigments, but that there's a reaction center in the middle, and that reaction center does have a molecule of, or a pair of chlorophyll A molecules in it. So that is that. Next, a lot of times in this unit, you'll see these little yellow zigzags of light. Photon, represents a photon, and a photon for us is just a tiny packet of radiant energy. So remember in the last unit, we talked about different types of energy, with radiant or light energy being one of them. And so a photon is typically represented by a yellow zigzag like you see there. Okay, then we have our equation for photosynthesis. As we go through this unit, you'll see that this is not much memorization as it is reasoning out. Because probably all you had to know in your Biology 1 honors class, you had to know a little bit about the light reactions and the Calvin cycle, and you may have had to know where it took place, what goes in, what comes out, where it happens. You still got to know that for here, but we do go into more detail, especially with electrons and hydrogens, which you'll see a little bit on the next slide. So in review questions today, I asked you what things plants need to survive. Well, they need water and they need light energy. Sometimes some equations will show light energy along this arrow. I tend to show it as a reactant. And plants also take in the opposite gas than we do. So since we take in oxygen, they're going to take in carbon dioxide. And then what they produce is glucose and oxygen. And so then you have those big numbers out front. Those are your coefficients, I guess. What they call them, superscripts. These are subscripts, but you have these coefficients out front. And so the way I kind of think about it is everything has a 6 in front of it, except glucose, because it's already got its 6 carbons. You don't want to make it 36. And light energy, because we don't put a number in front of words like that. Okay, but everything else has a 6 in front of it. Reason it out. What do plants need to survive? Sunlight, water, and they take in the opposite gas than we do. 6 goes in front of everything except light energy and glucose, or else you'd have 36 carbons you're messing with there. On the fun part. So I don't think any of you are taking AP Chem, or at least not this year. This is one thing that definitely is different than what you would have done in a biology honors class. I kind of warm you up to this concept in this unit, expose you to it a decent bit, and then when we get to respiration, we talk about everything that gets oxidized or reduced. And I show you how to pick that out. It's not a mystery. But in this unit, I don't dwell on it quite as much. We talk about it a little bit. But in the next unit, it hits you really hard. So you've got to get this concept. Okay, oxidation refers to the loss of electrons. Reduction, I know that sounds weird, but reduction is the gain of electrons. So why would that be? Well, since electrons have a negative charge, if you're gaining electrons, you're kind of reducing your overall charge on the molecule. So that's why reduction is gain of electrons. But if you're like me, you might have trouble keeping this straight. So luckily there is a way to remember that. The loss of electrons is oxidation. Loss of electrons is oxidation. The gain of electrons is reduction. Gain of electrons is reduction. And so you just remember that Leo the lion says grr. And then you'll never mix them up. Might take you a second to think about it, but you won't mix them up. And so then we're gonna, we need to talk about how to track this. Electron, I know you think, well, this isn't chemistry. Why are we talking about electrons? I know, but we have to do it because we have to know about things getting oxidized and reduced in photosynthesis and respiration. But electrons are still a little abstract for us. And so we are going to track electrons by tracking that molecules or that compound's hydrogens, because we are specifically following the electrons of hydrogen. And so to know if something is gaining or losing electrons, we are going to look at, is it gaining or losing hydrogens? And so that's why we have that equation, the photosynthesis equation at the bottom of the page, so I can help demo this for you. All right. So this is the same old photosynthesis equation I showed you before, and I went ahead and connected the compounds, the molecules that I want you to compare. So first we're going to look at carbon dioxide. Remember, we're going to track what the electrons do by tracking hydrogen. So does carbon dioxide have any hydrogen in its formula? No. Look what it's going to become. It's going to become glucose. Does glucose have hydrogens? Yes. So in going from CO2 to glucose, have I gained or lost hydrogens? I gained. And so what is the gain of electrons or the gain of hydrogens called? Reduction. So this is a reduction step. And we say that CO2 gets reduced to become glucose. All right, then let's look at your red arrow. The red arrow is water. Does water have hydrogen?
Yes.
Does oxygen have hydrogen?
No.
No. So we are going from water to molecular oxygen. We are losing hydrogens, and therefore we're losing electrons. The loss of electrons is oxidation. So along this red arrow, we're going to write the word oxidation, and we're going to say that, if I can label it, we're going to say water gets oxidized to form, you go, Lenny, to form oxygen. Now, the way this is going to come across in the next couple days, we can talk about overall, because this is the equation for all of photosynthesis. We can talk about what gets oxidized, what gets reduced. It is not something you want to memorize. It is something you want to be able to reason out by looking at it. Because in the next unit, there's going to be way too much to memorize. You can't possibly do it. You just have to understand what the two processes are and be able to think through it. And then we also talk about what gets oxidized and what gets reduced in a certain part of photosynthesis. And so that's what we're going to end with today, are the two parts. Because a lot of people think, oh, the light reactions are photosynthesis. No, the light reactions, or what they call the Z scheme, is just one part of photosynthesis. They are not synonymous. And so that's why you have your little flow chart on the next page, so we can go over what the different parts of photosynthesis are. So two major processes or parts of photosynthesis. You've got the light reactions and the light independent reactions. The light reactions can also be called the light-dependent reactions. The light-independent reactions can also be called the Calvin cycle. They are not called the dark reactions. That is an antiquated name. And if you've heard the term carbon fixation is one part of the Calvin cycle. So those terms aren't really interchangeable, but Calvin cycle and light independent should be interchangeable. All right, and in the bottom box for each one, we need to know just mainly what occurs during these parts. Why is it so important? Well, what you're doing in the light reactions is you are taking the radiant energy from the sun and you are converting that into chemical energy in the form of ATP and NADPH, okay? So solar energy or radiant energy is converted into chemical energy in the form of ATP and NADPH. Notice I didn't say anything about sugars there. Sugars are not produced during the light reactions. They are produced during the Calvin cycle. So that's why photosynthesis comes in two parts, because in this first part, you are converting light energy into chemical energy in the form of ATP and NADPH. And then in the light independent reactions, or in the Calvin cycle, you're going to turn around and use that ATP and NADPH in order to make sugars. So to me, it's kind of like, hey, it's my birthday. That's the birthday money I got. I'm going to turn around and spend that birthday money and buy something for myself. You make this first, but you have to use it in this part. If you don't make this, this doesn't happen, because you have to have that ATP and NADPH that you made in the first part in order to make sugars in the second part. So this is what I want you to work on. I want you to work on it by yourself. I'm giving you a Scantron to fill in, and then I'll tell you, because there's one question that you can't do on the Scantron. So I'm going to tell you how that's going to work. This is taking some thinking. Like I said, this is a hard unit, and I've done a good job explaining absorption spectrum to you, so I expect that you can apply that knowledge. And I'm also going to show you an image in color up here that may help you with, I think, the second page. All right, so there are 25 questions in this packet. However, if you look at the bottom right-hand corner of the first page, you will see that 10 is open-ended. So you will not bubble anything in for number 10 on your Scantron. You will write the answer to number 10 on the back of your Scantron. Okay, so I would go ahead and put that now, like make yourself a note, write number 10, write out. You are not going to bubble anything in for number 10. You are going to do number 10 on the back of the Scantron. And then the other thing I was going to show you is this, because this is what's on your second page, except yours isn't in color. And there's some pigments that you've seen, like you've seen chlorophyll A, you've seen chlorophyll B, you've seen carotenoids, but you haven't seen phycoerythrin and phycocyanin, although if you've changed ink in your printer, you've seen cyan before. Cyan is blue, erythro is red. So if that helps you with the second page, if you want to just tell yourself, okay, this line is red, this line is blue, that's fine with me. But I want you to do this by yourself. You can use your notes, use whatever. I just don't want you using each other. So I will leave that up there for a few minutes. If you want to color code that line, you certainly can. Sure, go ahead.
right, guys, finish that up for homework, please.